All right, great job, ladies. So our next speaker tonight is Skye Jacobson, and she is a biochemistry and a chemistry major, and she works with Dr. Rogers in the same department. And her presentation is going to be on endothelial nitric oxide synthase, or eNOS, crystallography and HPLC studies. Skye?
Hello. So this will be a slight change of pace from the last conversation, but I've been, since the second semester sophomore year, I've been working on looking more into what exactly eNOS is. So what is nitric oxide synthase? Nitric oxide synthase is an enzyme that you can find in many mammals, especially, and you can find in human beings, and it's responsible for the catalytic turnover of L-arginine, an amino acid, into L-citrulline, which is an amino acid derivative, and nitric oxide. So nitric oxide is a really important gaseous signaling molecule that's found within your body. And it has a lot of important functions, but the one that is mostly associated with the version that I look at of nitric oxide synthase is vasodilation, especially in regards to your cardiovascular system. So, the next one. So in 1988, Dr. Fergat, which some of you may recognize because he's actually a Charleston native, he was one of 3 scientists awarded with the Nobel Prize in medicine and physiology for the discovery of the importance of nitric oxide in the cardiovascular system. So I just wanted to highlight him there because it's like a cool fun fact to share along with the research side of it. But my focus, eNOS, is nitric oxide is what is responsible for producing that nitric oxide. So there are 3 isoforms of nitric oxide synthase, but the focus of our research is on endothelial nitric oxide synthase, the one that has to do with the cardiovascular system. So the protein itself is a homodimer. So homo means same, dimer is 2. So it's basically just 2 pieces of the same protein stuck together. Each monomer consists of a reductase domain, which is responsible for supplying an electron, which I'll explain all of that later, and a heme domain, which has a little iron that's part of the structure too. So in order for this to be fully active, for you to have that initial reaction that I showed at the beginning, it requires 1 bound H4B, or tetrahydrobiopterin, per monomer. So what is that? Tetrahydrobiopterin, big word, I'm going to call it pterin, you can call it pterin. It's a necessary cofactor for the enzyme eNOS, and what makes it super interesting in the case of nitric oxide synthase is that in the human body, in every other instance, it donates 2 electrons, but for eNOS, it only donates 1 electron. So that's an interesting interaction. That's another cool part of eNOS, especially for me. But eNOS wouldn't be able to fully convert L-arginine to L-citrulline and nitric oxide without the use of BH4. So why am I so curious about this one enzyme? What's up with that? So we're kind of looking more at, a focus for my work has been on what happens if you decide to change the active site of the protein. So you just tweak 1 amino acid that's in the structure. Or what happens if you decide to use a pterin analog, so just the pterin, but with something different about structure, or a different substrate. So the NOA as opposed to the L-arginate. Will it still happen, or how will the active site change? How will it be impacted? So that's a big focus for our lab, is tweaking 1 section of it and seeing how it's impacted throughout the rest of it, so you can have a better understanding of how the protein works. This is a picture of the protein itself. It's like I said, it looks like you have 2 pieces literally that could overlap with each other. And then I believe this is the iron porphyrin, the heme section of it. But how can you examine these changes to see if something happens? That's been the main focus for me over the past 2.5 years, I'd say, has been on developing an HPLC assay. So you can do single turnover reactions to figure out if anything's actually happening, or if what happens when you change 1 component to the whole thing. So when I was saying earlier, analogs of the pterin. So we have access to 4-methoxy tetrahydrobiopterin. Once again, just call it pterin. I'm not going to call it full name. 5-methyl tetrahydrobiopterin and 3-methyl tetrahydrobiopterin. So these different analogs can have different impacts on the enzyme. So it's cool to kind of see what impact they have, and you can check what impact it has from crystallography and the HPLC assays. And then another thing is the use of the mutants. So in Chem 355, Dr. Rogers and Dr. Fox, another professor within the chemistry and biochemistry department, they worked with students on site-directed mutagenesis of arginine. So this was the process of actually changing the active site of the protein itself. So I have access to 3 different mutants of the protein, which we work on growing, and then getting to apply to these different assay techniques to see what kind of happens, or can the protein still work if you change this 1 piece of the structure. So I kind of wanted to start out with going through what the typical growth process looks like for protein. So for an eNOS growth, we have a specific plasmid that we've transformed into E. coli in order to grow in this whole entire process. So I'm just going to kind of like go through a highlight reel of what a growth kind of looks like. So you would add one milliliter of overnight E. coli growth, and we treat it with 3 different antibiotics because our plasmid's especially engineered so that it's resistant to these antibiotics. So we can target it and make sure that only that protein is growing, or only that E. coli is growing. And you grow to an optical density of 0.6, 0.8, and then once you reach that, you add, so this is a big word, diala. It's basically just a heme precursor to make sure that you're going to be able to produce enough to get you to the protein structure, and then IPTG, which is basically just sugar to help it keep growing overnight. So to start growing our like ideal protein. And then this is the protein extraction and purification. So it's a lot of stuff, but the highlight reel is basically the proteins in the cells. So it'll be in the pellet in the beginning after you do the main centrifuge the next day. So then we need to get it out of that. So you'll pour off the supernatant, so liquid on top, and you'll resuspend it in our like base solution, and then PMSF to keep it from splitting up the protein. You'll sonicate it, so get the protein outside of the cells, because it's in liquid now, so you can actually do that. And then we'll centrifuge it again. So now that it's outside of the cells, it'll be stuck in the liquid, so we can actually separate it from the other components of the solution now to get the target protein. And we do that using an HPLC, which is a type of liquid chromatography where we use a column. We use 2 different types of columns, and to isolate the eNOS, which has been engineered to have a particular affinity to that specific column, so that we can separate it out and use it for our HPLC and crystallography. So, and we can confirm that we have our protein through gels. So the columns. I think this chemistry is really interesting, so I kind of went more in depth with that. If you can go to the next slide. So we use 2 different columns. The first day we use a fast flow column. And another fun thing about the fact that we're using a plasmid is that we were able to engineer it so it has a 6 histidine tag on the end of it. So what that does is gives it a really big affinity for nickel, like loves itself some nickel. So the first day when you use a fast flow column, if you because for the little animation, it gets stuck to the column, and so that everything else, like the trash, will come out around it until you increase the concentration of imidazole in the solution, which is the structure here, which has a higher affinity for nickel, so then the protein will come off in later fractions. So that's like the initial separation technique, and then this is an image of what like the uncut protein gel would look like. This upper band is going to be that because it's a little heavier, so it'll be a little higher up on the gel. And it's just an image of the fact that it's a sumo fusion protein and the 6 histidine tag. So then next, how do you get rid of the histidine tag? Because we don't need that. So overnight, before using it for the HP column, or the high purity column, we put it on dialysis with ULP1, which is a special protein that can cleave the connection between the eNOS and the sumo protein. So whenever you put it on dialysis with that, it separates it out, and then when we put our eNOS through the column the second time, it'll go right on through. So it'll just, yep. And then now it comes off immediately, and it's also a lot cleaner because this column's more densely packed. It's high purity, so like cleaner in solution. So, and this is also something you can see again on the gel the next, that same day. Since it's cut now, it's lighter, so it's a lot lower down, and you can compare it to an uncut gel for that issue. It's all good. So this is just the process to get to the protein that we're going to use for anything else. The next step for me came with trying to figure out how exactly we're going to make this protein usable for the HPLC assays that I wanted to do. So something cool about eNOS, which I mentioned before, but I don't know if anyone remembers, but it has 2 domains. It has a reductase domain and a heme domain. So for the purposes of my lab and for what we grow, we only grow the heme domain of the protein. We don't grow the reductase domain. So what that means is this instant, like in the normal reaction, the reductase domain is responsible for reducing the heme on the protein so that it can actually be in a state where it's available to work in the overall reaction scheme that I showed you in the first place. So in a normal sense, when you click it, the electron's given to the heme, and you're able to go from iron 3+ oxidation to iron 2+. So now we don't have the reductase. Click it one more time. So in order to get around that issue, I opted to use sodium dithionite as an artificial reducing agent and use UV-Vis spectrometry to see whenever you had the reduced, to watch for complete reduction. So what that looks like is basically staring at a computer for a very long time and you would watch the heme peak, the heme Surrey peak on the scan. So when it's not reduced, it sits around 420, but when you have full reduction, it'll move over to 413 for a complete blue shift of the Surrey peak. Fancy words of saying it goes left. And then another way to note to see if you use too much sodium dithionite is if you add too much, you'll have a peak appear around 315. Very large peak, you don't want that. So basically, this was over like the first summer that I started working on this, and I was able to figure out a ratio between how much sodium dithionite I need per concentration of eNOS. Sorry, lots of words. So that was the first kind of hill to tackle with making up an HPLC amino acid assay. But the next was actually looking at the single turnover experiment. So the idea for this was adapted from Hirschman's experiments with inducible nitric oxide synthase. But the premise was just, since L-arginine, NOAA, and L-citrulline have very slightly different R groups on the end, you can differentiate between them to try and figure out, using retention times on an HPLC, if you have some, if like the reaction is able to go full course, so all the way to the end product of L-citrulline, and by hence nitric oxide, halfway, so to NOAA, or not happen at all, L-arginine, which is exactly what I wanted, because I want to see if I tweak one thing, am I able to get to the end? Am I able to get halfway? Like, what's going to happen? So in order to make these amino acids stick on the column for longer, though, since they are very similar and they're also very hydrophilic, like they love themselves in water, the first thing that had to be done was derivatization of amino acid products. So what that basically means is I use phenyl isocyanate in an organic reaction to add on a little hydrophobic tail to make it stick on the column a little longer, so that you can actually see some differentiation between all of the amino acids. So this was to prepare it for use on an HPLC, and then I had to do standard runs of just the amino acids by themselves to get where their retention times would be, which is kind of what I've been doing more this year. So for the HPLC, we're using a C18 column and a diode array direct detector, which I also like to call it DAD, so it's just funny to call it DAD. So what that basically means is you can scan it at 2 different wavelengths. So like, let's say I want to like examine it at one wavelength, but I know something else is going to pop up at a different one. It's good to hold it for both of them so you can get an idea of what's going to look like total, like overall. And then these are my solution conditions. But basically from this year, I was able to figure out a retention time for my L-arginine standard of 8.322 and a retention time for my L-citrulline of 5.2 based on like just continuous trials with it. And for the whole thing, I do just a elution hold. So I don't change any concentrations. I keep it the same the whole time. So this has been kind of the progress of now is the HPLC. So for conclusions and for further work, I'm hoping that the lab continues on the HPLC trials with the native eNOS heme domain with use of an internal standard in order to solidify the technique a little more. And also some things that I was able to do this year were adapt the protein growth and isolation techniques and determine retention times for L-arginine and L-citrulline. And then going forward, we're also working on the development of a colorimetric assay for nitric oxide synthase with a new device, which is super interesting. Something I'm getting to stay over the summer to work on a little bit more, thanks to Embry. So I would like to say, I would like to acknowledge my research advisor, Dr. Amy Rogers, my current lab mates and past lab mates, Dr. Fox and past lab members. I have a lot of acknowledgements. Dr. Corey and Ginny James, because they helped me figure out how to use an HPLC. Dr. Barker for all the organic help, Dr. Giuliano and Dr. Forsyth for all the help with HPLC and like methodology, the Department of Chemistry and Biochemistry, and funding from ERCA CERT, Embry, and SSM grants.
Awesome, thanks, Skye.
Okay, Skye, let's see here. Could this be used for E. coli study? So I guess could you talk a little bit about kind of the real world applications or where this work could potentially lead to in the future?
Okay, so in terms of other applications, in terms of the HPLC amino acid assay, if we get good standard times for these amino acids, it could be applied for other people's other conditions or other experiments where they might need to look at intermediates of amino acids. In terms of E. coli more, so we use E. coli to grow the protein in the first place through the use of a plasmid. I wouldn't necessarily say we look at E. coli as much, but yes, that is the best I can answer that question. If that's helpful.
I have another question. That nickel column approach to separate the compound, that's pretty cool. Is that a common approach?
Oh, for the histidine tag on the end of it? So whenever you have a plasmid, you can kind of, it's like fun with science. I think it's just like engineering with science. So the plasmid is set up so that you can add it adds a 6 histidine tag onto the end of it, so that you can make the nickel column viable for doing that type of separation. So you can get a cleaner purification in the end. So the ULP1 is like specifically engineered for that sumo protein too. So yeah, I think it's commonly done across like research for this type of thing, because it just helps with isolating.
Gotcha.
Yeah.
Scott, can I ask a question about the mutant proteins?
Yes.
So there was what, 4 targeted mutations or 3 targeted mutations?
3 targeted.
Exactly. So based on those mutations, those point mutations, and what we know about this enzyme or other similar enzymes, what do you think the impact of those mutations is going to be on the reaction rate?
So that is a great question, and that is what we want to look at. In terms of the proteins, I've been able to, this year I was able to, we have crystals with the histidine, so the R367H mutant version of it, but we're still waiting on the analysis for that because we use the synchrotron time at MUSC from their crystallography lab, but they have to do the workup for us. So it's been kind of like a pushback and forth for that one. But we haven't gotten to look in terms of use for the other 2 mutants yet. But I don't know. That's like what the fun part is.
Well, I just didn't know if you had if you had any information based on what we know about other similar enzymes, or if were they directed in mutations, or were they random mutations?
They're directed mutations.
Yeah, so I'm sure they were made for a reason.
Yeah, it had to do with, like on that original slide, it had to do with the proposed active site structure for it. So I think based on where the arginine is, what it was interacting with on the L-arginine, which, funny. I think with what it was interacting with in that site, it was just, if you change it from like that charge to non-charge, what impact it would have there. So I don't know.
Is there any link of those mutations to any, I don't know, human genetic mutation?
Not that I know of, but there might be something in the literature. That's always something I can look into and get back to you on.
Cool. All right, thanks, Skye. Oh, was there another question? No.